This is Life Sciences Paper 1 from November 2022. We're going to be starting off with Section A, Question 1. Question 1.1 is always in your paper multiple choice. And remember, the instruction for that is that various options are provided as possible answers to the questions. You are going to select the correct answer and you only write down the letter. You don't write down the whole word, just the letter A, B, C, or D next to the question numbers in the answer book. And I want you to promise me that you're always going to write them underneath each other. You don't have to save space on your exam paper. You just write them one line underneath each other. That is what you're expected to do. Okay, so let's start then with our very first question, which says, which one of the following maintains the shape of the eyeball? And you're given four options: the cornea, the lens, vitreous humor, and retina. What I would do in terms of a strategy for answering a multiple choice is, before I even look at the distractors or the options, I would try and answer this with the knowledge that I already have. So remember that our eyeball doesn't have bones or any support structures inside it, and yet it maintains a spherical shape. So you should already have in your head a possible answer. Let's help ourselves by looking at this picture of the eye. The things that are mentioned in our options are cornea. What else? Lens, vitreous humor, and of course the retina. We know that the retina is the part of the eye where we have got the nerves, the photoreceptors that are going to convey information to the brain. We know that the lens is there to focus the image onto the retina. We know that the cornea is the transparent part of the outermost layer, the sclera, and the front of the eye. It's known as the cornea because it is transparent, and that leaves us with the vitreous humor. Now, vitreous humor is in fact a liquid, a jelly-like liquid, and it fills the back chamber of the eye. The reason that it is transparent is so that light can move through it without being distorted. But the other purpose of that vitreous humor is to act almost like a hydrostatic skeleton and maintain the shape of the eyeball. So if we go back to our question, we know that the answer for 1.1.1, the answer should be C, vitreous humor. I hope you got that. The next question is also associated with the structure of the eye. And this question asks the choroid, and straight away I want you to think, let me not look at the options. The choroid belongs to the middle layer of the eye. We've got the fibrous tunic on the outside, which is the sclera. Then we've got the choroid, and then we've got the inner tunic, which is the neural one, the photoreceptor or the sensory tunic. So what do we call that middle tunic or the middle layer? We actually call that the vascular tunic, because the point of the choroid, if we go back to our little diagram, here's the choroid, is, there we can see it's in the middle, sandwiched between the retina and the sclera. That choroid contains all the blood vessels. its name is the vascular tunic, so it should be fairly self-explanatory that there we're going to find the blood vessels that bring oxygen and nutrients to those all-important photoreceptors and make sure that they can get their wastes carried away as well. So let's now look at the options. The choroid is richly supplied with blood vessels. I agree there, but we don't stop there. We make sure that there's not a better option. The choroid contains photoreceptors. We know no. That is the job of the retina. Refracts the light rays? Absolutely not. That's the job of the lens. Sends impulses to the brain. That is the job of the optic nerve. So we've made sure that our first decision is in fact correct by checking on the other options that were available. Once we're sure, we are going to write on our answer books 1.1.2, and the answer there is A. Grade 12s, I want you to remember that when you write your multiple choice A, B, C, or D, please make it as clear as possible what letter you are writing. Don't do the A like that. Make sure that they are clear, unambiguous capital letters. You don't want the examiner or your marker guessing what your answer is. Make it as obvious and as clear as possible. Moving on, 1.1.3. Which one of the following occurs immediately after fertilization? So we should have in our mind something like this diagram. And this diagram takes us through all of the processes from ovulation through fertilization all the way to implantation. So our question asks, which one of the following occurs immediately after fertilization? So we are focusing on events after fertilization. Let's go to our options. The blastula, which is a hollow ball of cells, is formed by meiosis. Now, yes, the blastula is indeed a hollow ball of cells, but it most definitely is not formed by meiosis. It's formed by mitosis. All right, so after fertilization, we have mitosis continuing all the way around until we have a blastula that is many, many cells large, but all of those cells are the product of mitosis. So getting back then to our option A is definitely wrong. The morula, which is a hollow ball of cells, is formed by meiosis. Let's go back to our picture. This is what we call the morula. It's the Latin name for mulberry, and that's exactly what it looks like. It looks like a little berry. But the same as the previous option, the morula is not a hollow ball of cells, and it's not formed by meiosis. It's in fact formed by mitosis as well. So B is not a good option. The blastula, which is a solid ball of cells, is formed by mitosis. The blastula is not a solid ball of cells. Remember, it's a hollow ball of cells, but it is formed by mitosis. But this part of the option makes it incorrect. So we better hold out for option D. The morula, which is a solid ball of cells, there it is down there, solid ball of cells, is formed by mitosis. Absolutely. And so that leads us then to come to the only possible answer for 1.1.3. We are going to write D. Moving on, question 1.1.4. On a hot day, and immediately you know if we're talking about hot days, hot water, cold water, we are going to be linking our question to thermoregulation, how the body of a mammal controls its temperature. Regulation controls the temperature. So let's see which option makes the most sense. We're talking about a hot day. Less blood flows to the surface of the skin. Now think about it. If less blood flows to the surface of the skin, we're going to have retaining heat in our body because the blood doesn't go to the surface to radiate that heat out. So we've definitely got a problem here. I would say it's more blood flows to the surface of the skin so that your body can automatically get rid of that excess heat. The sweat glands become inactive. That's a problem. You know that on a hot day, you certainly sweat a lot more because as the sweat comes to the surface of the skin, it evaporates. And in that evaporation, it cools the surface of our body. So far, I don't like A and I don't like B. More blood flows to the surface of the skin. So that is giving us A, but it's giving us more. And we even wrote the word more when we were interrogating A. So I like that option, but let's just check option D. Vasoconstriction takes place. We're looking at vaso, which is talking about our blood vessels. And constriction means making narrower. Remember that our blood vessels can dilate or they can constrict, depending on how much blood needs to or is going to flow through those vessels. So vasoconstriction means make much narrower blood vessels, and that means that less blood will flow to the surface of the skin. So we've already established that, in fact, D is a repetition of A, just using a fancy term rather than explaining what that term is. So that is incorrect, and our answer for 1.1.4 has to be C. Now we're going to look at 1.1.5. The normal site of fertilization in a human female is, so let's look at our diagram as well. Is it the uterus? Is it B, the ovary? Is it C, the vagina? Or is it D, the fallopian tube, which is also known, remember, as the oviduct? You can always use these terms interchangeably. So where does fertilization take place? Remember that after ovulation, fertilization is going to take place here, anywhere in the oviduct or fallopian tube. So the sperm would have swum in. Some would have gone up to the left-hand side, some to the right-hand side, and this is the area where fertilization takes place. So our option, the only option that is correct, 1.1.5, our option here is D. 1.1.6. Which one of the following best describes the events of accommodation when a person is viewing an object that is less than 6 meters away? Before we look at our options, let's make sure that we understand all the terminology and exactly what this question is asking us. When we talk about accommodation, we mean changes in the shape of the lens in order to bring about accurate focusing of the image on the retina. So this question asks us for viewing an object that is less than 6 meters away. In other words, this is something that is close or nearby. So you reading something, you looking around at your desk, not you sitting back and looking at a television screen, but rather you sitting and looking at your Tenfold Education app on your digital device. So we're talking about close vision, not something far away. And then we have all these various options. Grade 12s, this can be very confusing. And it's not difficult to understand. You just have to picture in your mind what is happening before you look at your objects or your options, should I say. So let's remind ourselves of how accommodation or the change in the shape of the lens actually takes place. So this picture is focusing on close by objects, which is what our question asked us all about. And there are going to be 4 things that happen. The first thing is that our ciliary muscles inside the ciliary body contract. And what is the effect of that? It pulls the sclera inwards. And in pulling the sclera inwards, these little suspensory ligaments go slack because the sclera, if you can think of it, has been pulled inwards, closer to each other, or the ends of the sclera are now closer to each other. And that means that our ligaments are going to go slack, and it's going to cause the lens to adopt a rounder shape because there's not a lot of tension on that lens. Everything's collapsed inwards a little. Focusing on a distant object, we're going to see equivalent 4 actions that are going to take place, but they're going to be the opposites of what we saw for the near object. So instead of the ciliary muscle contracting, we have that ciliary muscle relaxing. And when it relaxes with the ciliary muscle, the sclera that was pulled close relaxes open, moves moves outwards. So as a result of the ciliary muscle relaxing, we're going to see the sclera moving outwards, and that is going to pull those suspensory ligaments taut, or you can see the difference, much tighter. And that in turn is going to pull the lens flatter or longer. I think that you need to be very sure that you understand those 4 actions in the eye and that they are exact opposites of each other. What happens for near vision? What happens for far vision? The fact that the same actions are going to take place, but they're going to be opposite each other. So remember our question asked us about close vision. Let's look at the options. The ciliary muscle relaxes. Let's go down to our explanation here. And we said when we're focusing on a nearby object, our ciliary muscle contracts because it's got to pull that sclera inwards. Okay, so that alone already tells us that option A is incorrect. So if option A is incorrect, option C must also be incorrect because we can rule out the ciliary muscle relaxing from any of our options. And so we're left with B and D, where we know the ciliary muscle contracts. If we look at B, the ciliary muscle contracts, causing the suspensory ligament to slacken. Let's go back to our picture. Yes, when the ciliary muscle contracts, it pulls the sclera inwards and our suspensory ligaments slacken. So that one is correct. Going down to D, we see that we've got the opposite, tighten. So I'm looking at probably B being our correct answer. Let's check it right to the end. Contracts, suspensory ligaments slacken, tension on the lens decreases. What do we see here? The lens becomes rounder because tension has decreased. It's not pulling that lens very flat. And so most definitely then B would be our option. Another way that I like to remember this to help trigger that memory is that when you're looking at something close by, your lens is rounder, almost like that C. When you are looking at something a long way, our lens becomes longer. That might help you remember the set of 4 conditions that needs to take place as well. So our option B is most definitely our answer to question 1.1.6. And now, still a multiple choice question. We read that 2 of the questions that follow are based on this diagram of this neuron. Let's not even look at the questions. Let's first interrogate this diagram. What are we looking at? We're looking at a neuron. What kind of neuron are we looking at? Well, we always go to the cell body and we work out how many projections are there coming out of the cell body. And here we definitely have 1 projection coming out of the cell body, which means we are looking at a unipolar neuron. And our unipolar neurons are associated with sensory mechanisms. So these are the neurons that collect stimuli, all right, which is then going to be passed on either to the brain or the spinal cord for processing. So we've looked at the diagram and we've satisfied ourselves that we've got 1 process coming out of the cell body, which means we're looking at a unipolar neuron or a sensory neuron, and at one end we have dendrites that are going to collect an impulse, and at the other end we've got an axon that's going to pass that stimulus on to some other neuron. And how do we tell the difference? Your dendrites tend to be pointy. Our axon terminals have little buttons on the end. All right, so now we've orientated ourselves. We know everything there is to know about this diagram. Let's look at the first question. 1.1.7 says the axon is represented by structure 1, 2, 3, or 4. Let's go back here. Here is 1, and we know that's looking at a dendrite. Here is 2, that's pointing to our cell body. Here is 3, which is pointing to the myelination of the Schwann cells, which are a type of glial cell that's wrapped around the length of the neuron in order to support it and to speed up propagation of that impulse. And here we have 4, and it's pointing directly to this part of the nerve, which is associated with the axon. So the axon is represented by structure 4, and our answer then must be D. 1.8, which labeled part affects the speed of impulse transmission? Going back to our picture, we've already spoken about it, the myelination with these little nodes of Ranvier or little gaps in between the Schwann cells helps to cause our propagation of the impulse to jump from node to node, which makes it much faster. Remember, if you walk and then if you run or jump, you get to a place much quicker. So our myelination, which we said was, or which is, number 3, is what's going to speed up our impulse transmission. So our answer for 1.1.8 is going to be C. All right, we have the last 2 questions that are multiple choice questions, and once again, 2 questions referring to a diagram that shows an investigation done. What is that investigation? It's determining the effect of auxins, which is a plant hormone group, on tropism. Before we even get to everything, unpack all those definitions in your mind. We are looking at an investigation. We must anticipate then questions about maybe a hypothesis or an independent variable. All right? We're determining the effect or the consequence of auxins, and we know that, as I said, they are plant hormones, and we know from our studying that they are plant hormones that stimulate growth. And the type of growth reaction we're looking at is a tropism, which is a movement of the plant, a growth movement of the plant in response to a stimulus. Okay guys, so let's now, we've clarified what the question is about. Let's read further and look at the procedure, which is how that investigation was set up. The procedure was set up as follows. A pot plant, all right, imagine it in our minds, was placed on a stationary clinostat. The plant was exposed. Remember, sorry, let me go back to the clinostat. The clinostat is almost like a record player. It's the surface that you can put seeds on, or a pot plant, you can fix that onto it, and it's going to rotate or stay still, depending if you turn it on or keep it off. The plant is exposed to light from all directions, and the growth is going to be observed. So we're going to see what is happening to the growth of this pot plant. Why do I like to draw little pictures of the words in an example or in a question? For me, it's a way of saying this is what's happening here. Now, if you are a second language learner writing this paper, your home language could actually be isiZulu, but now you have to write life sciences in your first additional language or even your second additional language. Help yourself understand the language as best you can. The language might be English, but the diagram has no language. And it will help you to be able to make sure that you've picked up all the language clues and you understand pictorially or diagrammatically what's going on. So that's why I always like to interpret the question in a little diagram to establish for myself, right, here's our pot plant. It's on a stationary clinostat, so it's not revolving or turning around. We've got light from all directions, and we're going to observe the growth of the plant. All right. And then we're given a diagram. Does our diagram match my diagram? Well, it does, in that here's our pot plant, and here's our clinostat, but it said the pot plant was placed on a stationary clinostat, and I interpreted that as there's the clinostat with the pot plant on top of it. And now in the diagram, I see that the pot plant is actually at right angles to the clinostat. But other than that, everything is the same. And I know that my clinostat is switched off and therefore is stationary and not revolving. The results after a few days showed the stem growing upwards. Remember that we had light from all directions, and that means that the stem started growing upwards. Which one of the following is an explanation of the results? So now we've got to think of what would happen, the stem growing upwards, and we've got to look at our options carefully to see which one explains our options the best. A, phototropism, which is the growth response to a light stimulus, occurred because, so I'd agree, yep, phototropism occurred, but here's why. So let's look at the reason. Because the auxins moved towards the light, which inhibited the growth on the lower side of the stem. And that, to me, is faulty. We know that auxins do not appreciate the light. Auxins move to the dark side. All right, well, you remember that auxins have a dark side to them. They like not a lot of light. So when we had our meristem, and the auxins are produced here at the top of the meristem, they start moving downwards, away from the light. And if we place the light coming from one side only, we would see that the auxins are produced, and here's our light. The auxins move to the dark side of the stem. And they, remember, auxins stimulate mitosis and cell elongation. And so on the lit side, or the brighter side, we're going to see less mitosis and less cell elongation. So if we've got something shorter here and longer there, it means that our stem will move in terms of its growth pattern towards the light. So phototropism occurred. Yes, it did. Because the auxins moved towards the light? Absolutely not. All right, so I don't like A at all. Geotropism occurred because the auxins moved downwards, which stimulated growth on the lower side of the stem. Now that could sound reasonable if it were not for the idea that the plant was exposed to light from all directions. And so we must realize that our light isn't only coming from the top, but light is coming from the side. Light is coming from the bottom. And so if we were looking at a phototropic response, we would see this stem growing out that way. But we're not only looking at phototropism here. Remember that our clinostat is stationary, which means that we're allowing the force of gravity to affect our plant. Our plant has a top and a bottom. So now, whereas we thought this had to do with light, in fact, we see that it actually has to do with the force of gravity. It is a growth response as a reaction to the force of gravity. Geotropism occurred because the auxins moved downwards, which stimulated the growth on the lower side of the stem. So now we've got our stem, and geotropism means that we have the auxins, by the force of gravity, move to the bottom side of the stem, and the top side of the stem therefore has shorter or smaller cells, and that's going to cause our plant to grow upwards. So I quite like B. Phototropism occurred because the auxins moved away from the light. Yes, we know that that happens, which stimulated growth on the upper surface. Yes, that is correct, but we're not actually looking at phototropism here. Because our clinostat is stationary, and it said we've got light from all directions, we are removing unidirectional light as a possible variable. So in fact, the only thing that we have got affecting the growth pattern here is in fact geotropism. So although the statement is correct according to our understanding of how phototropism works, this statement is incorrect relating to the particular investigation that we're looking at. This is a tricky one. So C is not the answer. D says geotropism occurred because the auxins moved upwards, which inhibited growth on the upper side of the stem. The auxins moved downwards under the influence of the force of gravity. So we know after lots of reasoning that our answer for 1.1.9 is going to be B. Do you see that in your multiple choice you haven't only got straightforward, easy questions, you've also got questions that require a lot of unpacking. A control for the same investigation was set up by putting an identical pot plant on a rotating clinostat. So now our clinostat is switched on and it is rotating, which means that the pot plant is going to get light from all angles, but it's also going to get the force of gravity removed, because at any one time on a rotating clinostat, there's no top and bottom. That top and bottom is going to be changing all the time. And that's why it is the control. Remove the force of gravity and see what happens. So what do we expect? There will be no growth? No. The plant will definitely grow. The stem will grow upwards. Well, we've said that that's what happens with the stationary clinostat. The stem will grow downwards. Why on earth would it do that? If it didn't grow upwards, it won't grow downwards. The stem will grow horizontally, absolutely. So 1.1.10, our answer is D. 2 marks per multiple choice. That leads us to 20 marks.